All right, everybody, welcome to Shed Aquarium's Shed Talk on protecting Magellanic penguins in Patagonia. Friends, it is Friday, February 2nd, 2024, and we are starting to get trickling in here. I see we've got over 40 folks already signed up and logged in, which is awesome. I'm going to give folks an extra minute or so to navigate technologies. I know Zoom likes to update every once in a while, so I want to give folks an opportunity to really dive on in and get in here before we get started, because we have such a wonderful next 45 minutes to share with all of you. Really high level stuff. So I'm super pumped to have all of you members and donors here with us today, joining us on your lunch. Before we get started, I do want to set the table. Friends, as you notice, we are in a Zoom, which means that we are in a webinar, which means you won't be able to see each other. You'll be able to interact, and I'll talk about all those ways coming up. I just want to give it one more minute for folks to log on in and sign in and get anything updated that they have to get updated. But yeah, we're going to be really interactive. So feel free to participate as much as you are comfortable. There's going to be lots of ways for us to do so. But we're going to give it just one more minute. So happy that all of you are joining us today. If you are here and ready to get going, I have an incredible opportunity for all of us, because I just want to get started right now so that way folks, as they trickle in, they can see this screen and know how we're going to be engaging today. So we have this incredible thing called Mentimeter. We've been using it now for about a year, and it's really awesome, different ways to engage with us. So you can either use your QR code that you see here on the screen through your phone, or if you are joining us from a computer, if you're at the office and this is your lunch break, then please head to menti.com, enter that code that you see. Because the first question that I want us all to think about, because we're here to celebrate penguins and how we're able to use our research in our colony here to help these animals out in the wild. So while we're doing that, here are the folks that we're going to hear from today. We're going to be hearing from me. There we go. You can see me over there. I'm Brian. He, him pronouns. I'm the coordinator for the public engagement team. Super excited to be the host of all these Shed Talks. I absolutely love learning from the experts. And let's just start on the other side of me, which is Lana Gonzalez, who is the manager of our sea otters and penguins. Yeah, two hats for that one. I know, a lot of animals to take care of. And then, of course, last but certainly not least, we have our very own Frank Oliaro, who is the manager of our molecular and microbiome lab right here on site. And we're going to have a really awesome inside look at all of those. But let's just jump on in and get started with that first question, because I see we've got, well, almost around 100 people already logged in. And so we're just going to jump on in. But friends, if you haven't seen earlier, I'm going to share my screen to that Menti. But the question that I want us to answer, just off the top of your head, is how many penguins are in Shed's care? So if you visited us recently, you've probably already seen our penguin exhibit, and you may have counted them all. I like to say they're like Pokemon. You got to count them all. But here we go. I'm sharing that Menti screen. So, oh, wow. We've got so many incredible guesses going on already. I see 35, 40, 75, 25, 47. So I'm seeing a lot of late 30s into those 40s. Oh, I see a 60 as well. 25. I'm going to give this just another moment, because I love all this curiosity that's going on right now, because some of you are really close. I'm not going to give anything away. It could be higher than 60. It could be lower than 60. There we go. There's your over-unders if you're playing at home. But yeah, 43, 47, very specific numbers. I love this. All right. Well, let me just tell you the answer right now, because it's been a couple of minutes. We actually have 42 penguins in Shed's care. Is it 43? I've got Lana and no, it's 42. It's 42. That's right. There's no extra baby just yet. I know. Don't get Lana excited about that. But 42 penguins here at Shed Aquarium. And right now, I want you to introduce one of the greatest individuals that we have here that is going to be helping us. Oh, excellent. My screen was frozen for some reason. My apologies. But I want to now introduce our Shed expert, our resident penguin I'm trying to find another P word, but our penguin corraller, we can call her. But everybody, please join me in welcoming Lana Gonzalez. Lana, welcome.
Hi, welcome everyone. Thank you so much for joining us. We're so excited to tell you about our fun and exciting research that we've been helping with. So basically at Shed, we do have 42 penguins. We also do have 33 Magellanics. And 9 rockhopper penguins. Just checking, Brian. There you go. Just a match. So you can see the picture of our exhibit. If you've never been to Shed, you can virtually see what our exhibit looks like. We also do have had a robust breeding program in the past, so it does help us to understand the natural processes, the nesting that goes into breeding with penguins. Also with the fecal samples that Frank needs for the DNA testing that we're helping with in our research. We have accessibility to the colony. So if you know anything about penguins, you know they poop and they poop a lot. So being
Yeah.
able to have our access to penguin poop is really important. So we're able to move them to a spot, collect a sample relatively easily. Also, the knowledge of what our penguins are eating is very helpful because what you'll learn a little bit later in Frank's portion is that each type of food item has a different DNA marker. So if we know the penguins are eating capelin and herring or all these other different food types, that will really help Frank be able to know what to look for. So yeah, basically it'll help Frank discover the best technique to get his data because there's a lot of things that go into collecting fecal samples in the field and different conditions of, does it need to be chilled? Does it need to be room temperature? So being able to have those samples in-house that we know what the penguins are eating and then being able to play around with those storage processes is really helpful. So a little bit about the climate impacts for the Magellanics in Patagonia. You'll see on the map, Patagonia covers a pretty large section in Argentina. Shed's animal care and science team partnered with UC Davis researchers to determine the best ways to collect, store, transport, and analyze the fecal samples. But basically, there are different colonies throughout Patagonia. The one that we were, if you see the second map, Camarones, that city is just 45 minutes north of the Cabo dos Bahias colony, which is where I had the
Oh.
opportunity to help with some of the research last fall. But what they're finding is the Magellanic colonies that are in the northern part of Patagonia are doing really well. And the reason is there's a lot of anchovies, and anchovies are very fattening. Those birds are nice and plump and able to have their chicks and everything. And then the colonies in southern Patagonia, there are a lot of sardines, which are also a very fatty fish. So those colonies tend to be doing very well also. The Cabo dos Bahias colony is right in the middle of Patagonia. So they're a colony that sort of has to get whatever's available. So they might have to have some other types of fish that maybe not as fatty or as robustly nutritious as some of those other types.
Mm,
So they're
yeah.
kind of seeing that, maybe there might be a fewer amount of Magellanic penguins, but they might be traveling to follow the fish where the resources are, which also can be impacted by climates, with the temperatures of the ocean, those fish may be following that climate change. So also like we said, we're using the fecal samples to determine penguin diets, and that also helps us create an understanding of their movements. So what historically has been done in order to know what a penguin has eaten, traditionally a way to do that out in the field is they would have to analyze the regurgitation that the penguins would come back with after they were fishing out in the ocean. However, that is more invasive, and also, usually the penguins are coming back with that food to feed their chicks. So that's just a little bit less food that they would be able to provide for their chicks, which in a colony that maybe they're not doing as well can make a significant impact. So what we're really excited about is penguins poop. Penguins in the wild poop, penguins here at Shed poop. So it's just something they're naturally going to be getting rid of. It's not invasive. So we're really, really excited if this is a way that the researchers are going to be able to see what they're eating by the poop and not having to do those a little bit more invasive techniques. And then this
Yeah.
also helps us to leverage the animal husbandry with the molecular lab capabilities. And so these are a few pictures of our fieldwork in action. So Patagonia is beautiful, but the colony that we were at, so and one way that we would be able to know where to put down our fecal collection devices, which is really a fancy name for a piece of plastic, is we would look for a nest area that had a lot of fecal scattered around. So if you know much about penguins, at least the Magellanic penguins, they're pretty clean for the most part. So they're in their burrow or in their nest, and they'll usually exit the nest to be able to go to the bathroom, which keeps it nice and clean and tidy for the most part inside. So we're looking for a nest. You see on the upper left-hand side, a nest that has a lot of fecal matter. So then what we would do is put down plastic, and then you can see me with a big bunch of rocks. So what is very interesting about Patagonia, it is very, very windy. So if any little corner of that plastic gets a little bit of wind, it'll just blow it right away. So it was like a little treasure hunt. You had to look around for a lot of rocks so that we could place them around the edges of the plastic, to hopefully hold down every single corner. Nothing is exposed to be able to take that plastic away. And then we would just wait. We would come back the next day, see if there was some fecal matter. And also you want to make sure the nest had a bird in it, obviously. So sometimes if they're abandoned nests, they might not be coming back as regularly, but especially if they have eggs or have partners in there, they will be switching or coming back and forth. So there's a picture of Lauren and myself, another staff member that went last fall, and then a couple pictures of some fecal that is on the plastic that we were ready to collect.
Wow, that's so fascinating and a dirty job, but somebody has to do it, and I love that. All right, well, let's continue on, and let me shift my spotlight here because we have another question, and I know I've advanced the Menti, so I know some of us got a little extra taste of what the question is. However, friends, if you want to join that QR code or go to menti.com and add in that code again, because our question is we're about to hear from Frank, who is the person that analyzes the diets of both our penguins and, of course, the stuff that Lana collects out in the wild. So how do researchers like Frank analyze the diets of these animals? Do they do a direct observation of feeding? Do they do microscopic analysis of the stomach content? And I know Alana talked a little bit about some of the ways, but perhaps there's more. Do they do DNA sequencing of the fecal material, or do they do a little bit of both? Is it all of the above? So I'm going to give it just another couple of moments because we've had a lot of guesses already. And it looks like the majority of us are choosing all of the above. And just so that way I'm not the only one that has the view of that. I'll share my screen here so that way everybody can see just what's happening here. But yeah, we've got mostly everybody. We've got that one solo person who's playing against the odds, going to win it all. The DNA sequencing of fecal material, which literally is what Lana was talking about. So that could potentially be the winning ticket. All right, looks like we're standing still now for a couple of seconds here for all of the above versus that one for DNA sequencing of fecal material. And friends, you're all right because it is all of the above. So even if you chose one of them, it's every single one of them together. And if that sounds like a pretty dirty job as well, sequencing all of the poop, all of the diets, and bringing it in for testing and analysis, well, you're not wrong. And we have another incredible expert who's going to talk and walk us through all of the ways that Shed leverages what we have here on site and with the way that we're able to help these animals out in the wild. So let me introduce you again to Frank, who is our incredible manager of our lab. So Frank, take it away and tell us a little bit about how and what we're going to be using this poop for.
Well, thank you very much, Brian, and thank you, Lana, and thank you all for attending today. I'm so excited to see the tremendous number of people who have taken time to hear about some of our work and our research. So very exciting. Thank you very much for being here today. Before we get into some of the more nitty-gritty details about how we have analyzed some of these samples and how we have designed these techniques, I just wanted to touch on a little bit of history about the Molecular Ecology Lab, which we formerly called the Microbiome Lab. The lab was started in 2016, and it was made possible by a very generous gift from the Granger Foundation. The Granger Foundation has supported lots of big initiatives at the aquarium for many, many years. And this was one of them. And it was really dedicated towards trying to have a better understanding of what kinds of microbial communities we have in our water systems and associated with our animals, and how we can optimize those microbial communities to benefit the health of the animals. As you can imagine, an animal living in a closed recirculating water system might be exposed to many, many different types of microbes than, say, an animal in a natural ocean environment. So we were very interested in utilizing molecular DNA sequencing techniques to be able to analyze these full microbial communities. And this type of research has really grown in the last 10 years or so, especially with the advent of these new sequencing technologies, where we don't have to rely on particularly growing out a particular bacteria on a petri dish, for example, but we can actually just utilize their DNA sequences to identify all of them in sort of one big shot. So it's a very powerful tool. After the initial project goals were essentially fulfilled, and we published a few studies about freshwater communities in our some of our Amazon type environments and how those are affected by different things like UV treatment and CO2, as well as how things like water changes affect the microbial communities in our oceanarium. And then how do those water changes and changes in the microbes then in turn potentially have any effects on the animals that inhabit those exhibits. So we knew all along that these types of studies were going to continue to be very important, but that also these types of techniques can be leveraged for many, many other types of research questions, not only microbiology questions, but also conservation based initiatives, and using these tools for those types of questions has really expanded the scope of what we can do in the laboratory. So some examples of that, using these techniques, obviously, as we're going to talk about, for analyzing the diet of wild animals. One project we've worked on a little bit already has been in association with the Bahamian rock iguanas, who are actually visited quite often by tourists and get fed all kinds of random things. So the question from us there was, what are they being fed? And then how much of their natural diet are they consuming? So that was one of the initial questions that led us into more of this dietary DNA work. And that was really a lot of what my graduate work focused on as well, was analyzing diets of harbor porpoises using DNA techniques. Now, the sequencing technologies come a long way since those days. And we're now able to, like I said, sort of analyze an entire community of organisms in one shot, which is a really, really powerful tool. So the thought was, well, let's try to do this for this particular penguin colony that, as Lana mentioned, is in sort of this transitional zone in between these northern and southern colonies, that they might not be getting those hardy sardines or those hardy anchovies. And it's still somewhat of a mystery on what they might be eating and how far they might have to go to get their certain types of prey items that they're searching for. So really the first step for us was to utilize all of the tremendous samples that we can collect right here very easily and test out what kind of storage methods we would need to sort of mimic what the field conditions would be like. So what we did was take a series of samples from our penguins, then expose those into 3 different groups of storage. So we had a group that was stored in an ethanol solution, a very high concentration alcohol solution that we stuck into the refrigerator. We had another batch that was stored in that same ethanol solution and then just left to sit at room temperature. And we also had, as sort of the gold standard of storage for DNA work, is to freeze the samples immediately. So after we were able to get those samples from our own penguins, we went through a process of optimizing our DNA extraction techniques to be effective for penguin fecal samples. Little did I know that penguin fecal samples can be pretty challenging to work with in terms of DNA. First of
Oh,
all,
wow.
the DNA is degraded due to digestion. So it chops up the pieces of those long strands of double helix DNA. It also, penguins tend to excrete not only feces, but they also excrete urates at the same time, which can be inhibitory substances when it comes to DNA and what we call PCR or polymerase chain reaction, which is an amplification technique you might be familiar with now after the COVID
Yeah.
era. Very similar to how you can test for a particular virus using PCR. You can test for a whole range of different things using PCR. It's an extremely valuable molecular biology tool. And in fact, the inventor of PCR, Kary Mullis, won the Nobel Prize for this technique back in
Wow.
the early 90s when it was
Wow, wow, wow.
officially patented. So what you're looking at here is some of the data output from the samples that we collected here at Shed. So just a long story short, essentially the stuff you see on the right there is just lines and lines of command line computing code. And that's sort of how we whittle through all of the mixtures of DNA sequences that we get off of our sequencing machine. So we get this big batch of huge gigabyte full of gigabytes and gigabytes full of data, and we've got to whittle through that data and make sense of it all in some way. So what we do is kind of put it through this bioinformatic pipeline to, first of all, clean out any erroneous sequences that we get, sort of filter everything out so that we know we're looking at really clean, appropriate data that we can then make some inferences from. And after we've done that, we essentially compare all of our unknown sequences that we recover from these samples to databases of known sequences for all of these different organisms that we want to examine and see if they're present as a prey item. So these databases are free online. There's a huge database called GenBank that is
Oh,
run by
wow.
the National Center of Bioinformatics and Technology. And so we can mine those giant databases and create our own local databases for the organisms that we're most interested in, things like fishes, cephalopods, squids, octopus, and crustaceans that are likely potential prey items of the penguins.
Wow.
And so by leveraging those databases, we can then sort of blast our sequences against them and be able to identify what they are, and then in sort of a relative proportion and how much of each of those sequences is in each sample. So that's what you're seeing on the left side there of the slide is a graph of a series of 6 of our penguins, or 5 or 6, that we collected
Looks like
samples
6.
from. And all of those little indicators with the RT and the 4C and the frozen are the different storage methods that we tested for each of the different sample types. The one on the far right there is actually just a mixture that I created myself in the lab. DNA from different types of fishes and squids and crustaceans. So I could have sort of a positive control sample to test out with actual fish and prey tissue to validate the techniques. So as you can see, there's that big gap in the middle there where there's no data. And that was really informative to us because we realized that storing those samples at room temperature in the ethanol was not effective enough to preserve the DNA so that we could actually sequence it. So that was extremely helpful in designing the way that we could collect the samples in the field and store them successfully and transport them to Shed to be able to analyze them.
Yeah. Now, of course, this research is really helpful, but how are we using this research to help amplify and collaborate with our partners through this next year?
Yeah, a great question. And it's very exciting time at the aquarium. As many of you might know, we are under a bit of construction right now,
Yeah.
going through our heading towards our centennial commitment in 2030. The building is changing. Some paradigms are shifting, and it's a very exciting time here. So in late fall of 2023, we completed our brand new facility of the Aquatic Science Lab and Animal Hospital here at the aquarium, and it is absolutely gorgeous. This is a shared facility of the veterinary hospital, the water quality laboratory, the molecular biology laboratory and microbiology laboratory, as well as our conservation research team offices. So it was a very intentional and purposeful combination of these teams that used to sort of sit on separate sides of the aquarium so that we could have more crosstalk and come up with more organic collaborations with each other, learn more about what all of these experts do with their particular types of research questions, and how we can really help each other out with not only the facility, but also with ideas for collaborative research, not only here, but with our partners, either in the United States or abroad as well. So it's been just a fabulous addition to the aquarium, and we're super excited for what the future holds here.
Yeah, and to your point, it helps us here, there, and everywhere. And so now I want to bring Lana in to talk about that there and everywhere portion, because, well, we had the boots on the ground, or in the guano, as it might be a little more appropriate. So Lana, if you want to talk about the expanding research that we're going to be doing, not just in Patagonia, but beyond that.
Yeah, so some of the other field research that we were helping with while we were at the colony in Patagonia was nest monitoring. So even though there's about 7,000 pairs of Magellanic penguins at the Cabo dos Bahias colony, the researchers that we were working with were only monitoring about 200 nests out of those. And what we were looking for were, were there birds in there? Did they have eggs? Would they, certain amount of them have pit tags? So we would scan them. You can see the picture there, Lauren scanning to see if it's a bird that's identified. We'd note if it was a male or female. You can see I'm standing there. My pants are very poopy from laying on the ground, doing a lot of nest monitoring. It's hard to tell, but they are dirty. But we have also a crayon that you can mark pink or blue, so it makes it easy to tell when you monitor them the next day if it's the same bird, if it's a different bird. And then if there's eggs, is there one? Is there two? Because there is a lot of predation. It's just a natural effect of living out in the wild and having your chicks and eggs out there. But or maybe there was a new egg laid. So our researchers would measure them, see how big they were, and then they would be able to kind of see the success by the end of the season with their breeding. We also helped do a census of the colony. So basically, you can see at that lower right-handed picture all these little dots. So that's basically a picture of the colony in all these GPS locations that we would locate. And then we would do, you can see the other picture. It's really hard to kind of see, but there's a string. So we would walk in a circle and be able to note what types of nests. So if we see a bush nest, if we see a cave, we call it a cave nest where it's a little bit underneath the ground, or an open nest that's just out there. And then also how much use is happening. Is it active? Which means, is there a bird inside? Is there some use? There's fecal nearby, but maybe no bird. And then inactive, maybe some overgrowth of grass that doesn't look like it's being used. And then that indicates how the colony is changing over time. So maybe they're like, wow, last year there were a lot of coastal nests that were inhabited, and maybe there's changes, maybe there aren't, or those type of things. So that helps to, know a little bit more about how the colony changes over time. And then other studies, they do have some GPS trackers on their penguins at different times of the year, so they can see where they migrate, what they're feeding on, how they're diving, that sort of thing. And then also there's a predation study happening at the same time while we were there. So they have cameras that they can see, what predators are going in the areas. Also, if we would be walking through the colony and we were like, oh, we see a broken egg, maybe one of the gulls or the petrels, got an egg, and, we're able to be a predator in that area. So they would mark it down where it was, what type of, predators, if you saw different animals or birds like flying in the area other than just passing over, but just kind of circling, maybe searching for an opportunity to get a little snack, we would note all of those things. So all of those different studies happen at the same time at different times. Some of them are concentrated during the breeding season, and other times it might be like the predation study. I know that they were working on it outside of breeding as well. And then there is a potential for a shed adventure to Patagonia in the coming years potentially. So we just wanted to put that little bug in your ear. If it's something that you're interested in, maybe wanting to see penguins in their natural habitats, you want to cross that off your bucket list.
Wow.
There is that potential coming up. So stay tuned.
Yes. Oh my gosh. So many exciting things. And I see in the chat, and I also see some hands in the air, and perfect timing for that because, friends, now is the time for questions. So please follow that QR code, go back to Menti because we'll be able to get your questions on there. Also, if you have the chat pulled up, if you're on a keyboard, that's Alt+H. That's the hotkey for that. I'll be able to sort of facilitate questions from both of those potential areas. So if you have any questions for Lana or Frank, either about the field research or the research from our own hospital, please, now is your opportunity. We have at least another 15 minutes or so for questions. We were pretty good on timing, which is pretty awesome. Very much so. So everybody, it will give you just a couple of moments to type those questions in, either through the chat or through the Menti. But if nobody has any questions, that's perfectly fine too, because I know I have several for Frank and Lana, but nobody needs to stay for those. So really, my first question for both Frank and Lana is about how you fell into this work. Did you always know? So for Frank, did you always know that you wanted to go into research biology, microbiology? And Lana, did you always know that you wanted to go and work with penguins, not just in a zoological or an aquarium setting, but even out in the wild? So whoever wants to start first.
Yeah, I'll jump in, Brian. Thanks.
Yeah.
So yeah, I always knew that I wanted, well, not always, I guess, but when I got into my started my undergrad, I didn't absolutely know what I was going to do. I had a couple ideas. I thought, I like science, I like math, I also at the time liked architecture a lot, so I was kind of
Oh, wow.
undecided. But one day, it's this kind of a funny story, but I'll tell it anyway. I was actually working as a parking lot attendant at a restaurant, and as you can imagine, that can get pretty boring after a while. You're sitting in the lot for hours and hours and hours. Now and then, you might have a car pull up, and you have to, help somebody. But spending that time, I started actually just observing an ant colony every single day,
Oh,
and just
wow.
watching this colony really helped me pass the time, and it was extremely interesting. So that kind of changed my path, and I decided that I really wanted to focus on biology and animals, and animal science, and that was what really led me down the road towards research biology as well as microbiology. But um, I have always also had a passion for marine mammals and cetaceans and really all aquatic animals as well. So
Wow.
I grew up in New Mexico and decided I needed to get closer to the ocean. So I ended up moving to California and doing my master's degree out there in San Francisco, where I studied, as I said before, harbor porpoise diets. And at the time, I didn't have any clue about any of these molecular techniques. I was thinking, oh, maybe I'll go and start sorting through these stomach contents, like Lana had been talking about, looking at things microscopically. And my PA or my advisor really introduced me to the power of molecular biology techniques. He was the head of the DNA sequencing lab at the university, and he said, rather than looking at them, why don't you try sequencing the DNA of all of them? And it really opened my eyes to this entire world of the capability and power and flexibility of these techniques. And ever since then, I've really never looked back.
Oh, wow. That's fascinating. Absolutely. And hopefully inspiring for some folks out listening here today. So Lana, yeah, if you want to take that, and then we've got some questions to jump into. This is awesome.
Sure, yeah. Like many people, I probably have a similar background. I have loved animals ever since I was a kid, watching those nature programs or seeing them at zoos and aquariums. So I really wanted to work with animals. That was the dream. And I really didn't know that much about penguins until I started working at the aquarium here, but that was one of my first animal groups that I focused on. And I really fell in love with them and just learning so much. I'm sure everyone can understand they each have their own quirks and their own little traits and things that they like and dislike. So, getting to know each individual animal and really building those relationships has been super reinforcing and rewarding for me in my job. So, I've been so happy to work with penguins.
Yes. Oh my gosh. And yeah, you do an incredible job with both the penguins and the otters too. We can't forget that you are the manager of both of those critters, which is pretty awesome. All right. So now let's jump into some real questions from the audience. Not like my question wasn't real, but uh, our first question is about volunteering at Shed to help with this research. So Lana or Frank, are there any ways that folks can volunteer with either of your teams or departments to help with this research?
Yeah, so we do have a pretty strong volunteer program in the laboratory. A lot of it is related to the water quality laboratory. As you can imagine, there is a ton of testing that has to go on every single day. There's a lot of water here, millions of gallons of water, and all of it needs to be analyzed very, very quickly to make decisions about how the life support systems are going to be run by our facilities team and also by our husbandry teams. So there are definitely opportunities in the water quality laboratory. The molecular biology lab, we do have some volunteers that assist with sort of routine lab tasks and things, and then also sometimes have opportunities to help with some of the DNA work. Right now, we don't have any openings for volunteers in that lab particularly, but I would just encourage you to keep an eye on the website. I know that a lot of opportunities are posted there, as well as other resources. Brian, you might be better at pointing people to those, but through the website, there are lots of places where you can look for these opportunities.
Yeah, for sure. Lana, anything that folks can do for your team?
Yeah, we don't currently have volunteer positions in our oceanarium team. We did pre-COVID, but we haven't
Mm,
restarted it after COVID. But we do occasionally have offer internships
yeah.
in different various areas of our oceanarium team. So I would definitely just keep an eye out for the website and different places that we might post that when it's offered.
100%. And to that point too, if you check the website right now and scroll all the way down to the bottom where it says jobs and opportunities, I believe we have several internships available now. So check the website and yeah, get involved, be active. And just by being members and being donors, you're already helping to continue Shed's mission and vision here, there, and everywhere. All right. So our next question will be for Frank. What have the results shown from the fecal analysis in Patagonia? What are the penguins doing up there or down there, I should say?
That is the best question, right? And the one that's driving all of this. To be honest, we have not completed any of the sequencing for the wild samples just yet. I am
Oh, wow.
currently actually working on processing those. So I'm hoping to have our preliminary data sometime in the next month or so. So stay tuned. We might be able to share some of that out after we've completed it. But really, this was more this past year was really more of a methodology testing period, making sure that we were going to be able to successfully analyze these samples. And so before I jumped into processing a bunch of them, I wanted to be sure that we
Mhm.
weren't going to risk losing any data by using a technique that might not have been effective. So that was really where Lana's team and really the team of our penguins helped us figure out the best methods to analyze these samples. It was, I have to admit, very, very challenging. It was one of the more challenging types of samples that I have worked with. But now, after working through all of those kinks, I feel really confident that we're going to get some really juicy data out of
Wow.
these samples. And I can't tell you how excited I am to get that first sequencing run done and actually start looking at some of the real data. So we don't have any info yet on exactly what prey species we can identify from those samples, but now I'm very confident and know that we're going to have success in obtaining those sequences. So more to
Wow.
come on.
Yeah, that's exciting. And also sort of a life lesson, right? A 1,000-mile journey starts with a single step. And so this was that single step to hopefully help influence the rest of those other 1,000 steps. All right. Well, Lana, we've got a question for you and only you because you have the inside scoop to this question. If nobody knew, over the past several months, last spring, our colony did grow by one. So we have a little baby rockhopper penguin, which was the first in about 15 years, I think. And Lana, you can correct me on that one. But the question is, How is our baby penguin doing?
Oh, well, she's not a baby anymore. So
Well,
penguin
for sure.
chicks, if you don't know, are full grown within 3 months. So she definitely
Yeah.
is a juvenile. She definitely is spunky, has a lot of energy, and is very curious. But Esla is our rockhopper chick. Yes, she's doing great.
Yes. Oh my gracious. Awesome. Well, oh, and it looks like we just had one more question pop in. So let me get to that one. This is for either one of you. What was the main difference in behavior of penguins in Patagonia versus in our shed's habitat?
Yeah, so what's really interesting about the colony out in the natural area where I was, everybody, it depends on where they are in their nesting time. So,
Mm,
a lot of times the penguins in our colony, a lot of times they can hang out together, they're in the nest together, but that's not really a realistic thing of what would happen with penguins out in their natural habitat. Usually they would come together for a certain amount of time. Usually the males would come first, kind of return, get the nest ready. The female would come, meet back. But then, they would breed and then they would have to go off and start fishing so that they were ready to come back and switch with their partner, take
mhm.
turns. We do tell our penguins here, I'm like, you have a very cushy life here. You have restaurant quality food. You don't have any predators to worry about. And you just have, healthcare at your fingertips.
Wow.
So yeah, it was really, really cool to see them in their natural environment and just see how similar they are in like behavior, different mannerisms like that they would do. We're like, oh, we would see that at in our colony as well. So even though,
Wow.
they have differences, but those inherent things are the same.
Now, Frank, I want to extend that question up to you because, yeah, you weren't there, you didn't, and you're not actively in the habitat. But with your research and with that fecal samples, can you get a sense of the behavior of those penguins, or is it simply just, well, here's the diet and here's what we do?
Well, one thing I'll say that's really interesting is even just visually, these
Mm,
wild animal samples are completely different
wow.
from samples that we've collected from our penguins. Obviously, our penguins get like Lana said, restaurant quality, delicious fish that any of us would eat sushi, right? But
Yeah.
um, the penguins in the wild, there's so much more variability, obviously, into what they might be eating, which results in a lot of variability of the different colors of the samples. If, for example, if maybe they're eating lots of a crustacean like a krill species that's very, very red in color, that might in turn have that coloration of the fecal sample, and or variations of the texture and what else we might find in there. So bones of different animals, um,
Wow.
little bits of pebbles, even some, little internal parasites that I found in some of them, which
Whoa.
is obviously not uncommon among wild animals at all. So there's just a really, really distinct difference, even visually, looking at these samples compared with the samples from shed.
Wow.
So behaviorally, I think that also illustrates that they're doing a lot of different things to try and get their meals. It's not all just a consistent, they all look the same, they're all eating the same thing. So when we get some of these data, it's going to be really exciting to look at the variability and then being able to associate that with some of the GPS trackers as well that Lana had mentioned that our
Yeah.
partner Patagonia are using. So we might know exactly the track that an animal took, and then we can associate that with what it ate and be able to use those types of metadata in association with the sequence data to
Mm,
really make some powerful discoveries, and then hopefully be able to make some conservation initiative decisions
yeah.
about how to help these animals.
Wow, that is incredibly important. Thank you for bringing up that conservation note there. And really quick, we've got one last question here, and this one is for Lana. And the question is, how is one of the world's older or potentially oldest penguins doing? And if you don't know, we're talking about Wellington, the rockhopper.
Yeah, yeah, he's doing pretty good. He's hanging out on exhibit, and in a few short months it'll be nesting season again. So the time in the penguin exhibit, it's always changing. They have breeding season, they have molt season, they have sort of their migration season when they swim a lot. So yeah, he's been chilling and having a good time out on exhibit.
I know it's great to retire with a diet of 5-star quality sushi, right? Yeah.
Absolutely.
Oh, I lied. We actually have one more question, and this one is for Frank. Having discovered that you get better DNA results from frozen samples, well, I guess this is for both of you too. How would you freeze samples as you're collecting them out in the field?
That's a fantastic question, and it's a very challenging thing to do, as you would imagine. That um, there's not access to a freezer right
Yeah.
next to the colony, right? And the trip to the colony, correct me if I'm wrong, Alana, but probably about an hour or so just to get from your camp to the colony, and then from there, that's even further to get to an actual lab or something where there might be cold storage capability. So that was really the challenge and why we were looking at, rather than just collecting the sample in a tube and not adding anything to it, we were discussing the alcohol preservation technique. That's a fairly commonly used technique for DNA preservation, but we really wanted to test it out before we set on that. And we've also actually in the past field season, Lana brought down another solution, a preservative solution for DNA called DNA Shield, to test that out and see how the effectiveness is between those samples as well as the samples collected in ethanol. So very good question, and it's
Yeah.
definitely a challenge for field studies where there's just not that type of cold storage or cold chain available, and it's also a challenge to get the samples from Patagonia up to Shed and stay frozen the entire time there. So that's another piece of it, is the actual transport time when the samples are sitting. So that's why we're looking at the preservative method, so that we don't necessarily have to keep them frozen, but we can keep them cool and probably still have an effective preservation of the DNA. One more thing before I go, I just want to send out a special thank you and a
Oh.
shout out to the Koipu Foundation, who has provided us a grant. We were able to obtain funding from them last year to support our work moving forward. And so we are extremely thankful to Koipu Foundation
Wow.
for their generous grant that they provided us for this work. So thank you.
Yes, awesome stuff. And really quick, just a follow up before we uh, leave Frank and Lana to go back and doing the incredible things that we're doing here. For that Wellington question, I didn't set it up properly. So Lana, can you just really quickly, life expectancy of rockhoppers in the wild versus, of course, Wellington being 30 plus years in human care?
Yeah, so basically rockhoppers in the natural habitat would live just into their teens, and he's more than double
Wow.
that age right now.
Yeah.
So, every day we have with him, we treasure, just as we know animals get older, but yeah, we're enjoying him as long as we can.
Yes, and we will continue to update everybody on Wellington's whereabouts, of course, if you follow Shed Aquarium on any of our social media. Well, friends, let's just take a moment and thank Lana, thank you, Frank, for joining us and being here and sharing your wisdom and knowledge with us. I know I appreciate it, and I hope that our members and donors do as well. And I cannot have everybody leave without thanking all of you. So thank you so much for joining us today and being a part of us. Your support really helps provide a crucial part in our vision, which is to see a world thriving with aquatic life, sustained by folks who love, understand, and protect it. And I know I got this question in the chat earlier, but yes, this presentation will be available via a recording, and you should look for that in your emails roughly in about 2 weeks or so. And I also want to remind everybody that if you check your e-news, there will be a member night registration opening up here rather quickly. And speaking of members night, yes, save the date. It's going to be March 21st, around 6 p.m. to 9 p.m. Oh, and we've got lots of folks in the chat saying thank you to Frank, thank you to Lana. So yeah, we're not the only ones that are appreciating you right now. Everybody on this call is. Also, please, as I mentioned too, stay tuned for your exclusive member night registration. And everybody, I'll give you the rest of your 10 minutes. Hopefully you enjoyed and learned something new from Frank and Lana. I know I learned something new every time that I host these. So it's a real honor and privilege to be able to share everything that is happening with you exclusively, our members and our donors. And we cannot do our mission or vision without you all. So thank you all so much. Please enjoy the rest of your afternoon, and please stay safe and have a wonderful rest of your weekend. Thank you all. And on that note, I will let you all enjoy the rest of your day. Thank you. Thank you, Frank. Thank you, Lana. Thank you, Diane. Thank you, Gabriel. Thank you, Dawn. Thank you, everybody in the chat. This has been very special. All right, everybody. We will see you next time.